do now is kind of finish up at least the basic structure of the respiratory system and work through the rest of the lower respiratory tract. So so far we've talked about the nasal cavity and down to the larynx. Really the larynx is dividing of that upper and lower respiratory tract. So what we're going to try to do now is work our way down the rest of the way and talk a little bit about the basic structures of all this. In reality, the anatomy is pretty similar throughout. We're going to have a mucosal lining, so some type of epithelium with some underlying connective tissue. As we work our way further and further down, we're going to see at the beginning there's a lot more hyaline cartilage, and as we work our way down, we're going to see less and less of that and a little bit more connective tissue as we go. At the top part of this here, we are going to be working with a lot of pseudostratified ciliated epithelium here. The other thing we see in the trachea especially is these C-shaped rings of cartilage. Again, the trachea is not doing anything in the way of exchange. It's really all about holding open that airway so the airway does not collapse. Again, C-shaped rings of cartilage, not a full ring. Part of that reason is that the esophagus is traveling right behind this. And we need some space for that esophagus to actually be able to expand when you're swallowing a bolus of food. And again, a lot of the stuff is held in place by different ligaments. There's some muscles that help to flex this when you are coughing to change a little bit of that. As you work your way down the trachea, it is going to bifurcate or branch at the bottom. That mucosally covered piece at the very bottom is called the carina. This is super, super sensitive. If anything hits this, it tends to trigger some very violent coughing at that one there. So again, if someone goes on the wrong tube, that is a lot of times going to really cause you to cough violently on that one. Tracheotomy is sometimes if there is a problem with this, that they will actually poke a little hole in the trachea to bypass some of the upper areas here, and can be a life-saving procedure if something's blocking your larynx or not. So this is kind of, again, showing you that trachea traveling down to that bifurcation. Like I said, the carina is going to be that area right at that branch point. Again, anything touches that, it is going to cause some really violent coughing to take place. And again, those walls, the mucosa is going to be that pseudostratified ciliated epithelium with a little bit of connective tissue underneath. Beneath that is the submucosa, which is going to be composed of areolar connective tissue, a lot of blood vessels, nerves, glandular tissue, lymphatics, a lot of cartilage in the trachea about keeping that open, and then some elastic tissue surrounding that is making up that adventitia, which is similar to a serosa, but because we're not in a serous cavity, it is not a serosa. A lot of times I refer to all the branching of this as the bronchial tree, and if you were to kind of take your respiratory tract and flip it upside down, it tends to look a lot like a tree. So you have this main trunk that is a trachea, and then it's going to branch out to smaller and smaller branches, and ends in those very small respiratory bronchioles that are going to be going to the alveoli, which, if you think about it like a tree, are a lot like leaves on that tree. So it goes into those first main two. When it branches those first two, that's what's called a primary bronchi. That is that first branch. There's going to be one to each side, left and right primary bronchus. We're going to see the right side tends to be a bit shorter and more vertical because the other one is kind of working its way around the heart, so it tends to go a little bit more lateral across there. But what that usually means is if you aspirate something, more likely than not, it's going to block that right side, not the left side, because it is a little bit more vertical on the right side versus a little bit more horizontal on the left side. These are then going to branch again to go out to each lobe of the lung. So you have 3 lobes on the right side, 2 lobes on the left side. These are going to branch in each of those. These are what we refer to as secondary bronchi, that then go and start subdividing to each of the little segments of the left and right lungs, which those are called tertiary bronchi. And again, they're going to continue to divide smaller and smaller branch points here till you start getting these very small 1 millimeter wide tubes that are going to lead into, like I said, the last part of the conducting zone, which is called the terminal bronchioles, which are going to end in kind of this dead end cul-de-sac of these alveoli into a respiratory bronchiole. So again, if you look at this one here, if you were to flip this upside down, it does look a lot like a tree with a bunch of branches. And you can see how it branches down and branches down. Bronchitis, a lot of times, is if you get some type of infection, be it bacterial or otherwise, that is going to lead to a cough large amount of coughing and stuff like that. It usually takes a couple of weeks. Chronic bronchitis is one of those that it's a much longer term one, and again can cause issues. And it does, I mean, in terms of just health in general, chronic bronchitis is not something that's an easy thing to deal with. One of the things we see as we work our way down, we are going to see a lot of these same structures throughout. So that epithelium, that cartilage, presence of smooth muscle. What we're going to see is in these upper regions where it's all about conduction, we're going to see that it favors one thing a little bit more than others. So a lot more cartilage further up, more smooth muscle, and less cartilage as we work our way further down. And you can see here the left and right lung. You can see the right lung on this one, 3 lobes to it. Left lung, only 2 lobes, and does have a little notch out of it. You can see how that left primary bronchi is a little bit more horizontal due to the fact that it has to work its way around the heart. So you just have a superior and inferior lobe on the left lung, a superior, middle, and inferior lobe on the right lung. And as we get into these main bronchi, they are going to have some incomplete rings of cartilage. As we get further and further down, like I said, to those bronchioles towards the end, they start to have way less, actually to the point of having no cartilage. Because they're so much smaller, the structure in the wall is able to hold them open. We start to see a lot more smooth muscle in these areas, which allows for that bronchoconstriction or bronchodilation to let more or less air into those exchange regions. And you can kind of see it right here. You can see C-shaped rings up here, these incomplete rings as we get to here, a little bit more incomplete as we get to here, and you see as you get down to those very small bronchioles at the end, the cartilage has completely disappeared. So further up, it's about making sure that conduction is happening. As we get further and further down, it's more and more about regulating how much airflow is in there, and again, the small diameter tends to keep them open. Again, bronchoconstriction is the contraction of that smooth muscle will close that off. Bronchodilation is, again, opening that up. And as we get down to the very end here, that's where you get to these terminal bronchioles, which are going to go to these dead ends, which are the respiratory bronchioles. And again, if you imagine this all over the branch points on all this one, it starts giving you that idea of how it actually does look a lot like a tree, where you have all these branches and branches and branches going down to these alveoli at the tip. So they act, like I said, a lot like the leaves on that tree. The other thing to kind of notice on these ones is those alveoli, they're going to be surrounded by a very dense set of capillary beds. Kind of looks like a netting around each one of those. Means that we have these capillaries sitting right on side of these alveoli, both of which are just simple squamous only. They give us these really thin walls for this diffusion to take place across. And again, we branch down, you get to the point that it's about in the lungs about 300 million alveoli, giving us the surface area of somewhere between 1/2 and 2/3 of a tennis court of surface area to have this exchange take place across, which is part of the reason we're able to exchange our gases as quickly as we are. And we're going to see in these alveoli, there's really 2 main types of cells. You're going to have your normal alveolar cells, which are just these squamous cells. The main thing they are doing is they're acting as that membrane that the exchange is taking place across. The other thing these do is they make that angiotensin converting enzyme we talked about with the vessels. The other cells that are in here are called alveolar type 2 cells or septal cells. These make something called surfactant, which is kind of detergent-like. And the main thing that this does, if you think about if you were to blow up a balloon and then let all the air out, that water vapor will start sticking the walls together. The surfactant in these alveoli keeps surface tension of water from causing the alveoli to collapse. The other thing we'll see in these is some dust cells, which are alveolar macrophages that are specific to the lungs. And again, lots of muscle surrounding a lot of this, so we can bronchoconstrict or dilate depending on what we need to. And you can see that netting surrounding each of those alveoli to allow a lot of exchange to take place if it is letting air get into it. And again, you can see it with these capillary, with the kind of color drawing here with the red and blue, which kind of showing that oxygenation from the deoxygenated blood. The last thing to kind of point out on some of these ones is just kind of pointing out how overall the diameter is quite large when we're up at the upper parts here, but that there's not a lot of branch points. And the other thing you can notice, cilia and mucus is very high at the beginning of this. As we work our way down, we start to see less and less cilia, less and less mucus, a lot more cartilage at the top, and as we get down to the bronchioles, very little cartilage, and then you can see smooth muscle. Not a lot of it at the beginning because we're not doing a lot of regulation of flow, but as we get down to these bronchioles where there's not cartilage, we start really regulating how much flow is going in or out. The other thing is just getting the sheer numbers of these. As I was kind of saying, you have one trachea, we get two primary bronchi. As we start working our way down, there's more and more of them, and by the time you get into the alveoli, it's on the order of that 300 million, which gives us, when we start adding up all the surface area of that, quite a large area of exchange taking place here. And again, what makes this happen so well is the fact that it's really the whole thickness that we have to diffuse across is only about a half a micron thick. We have a simple squamous cell butted right up to a simple squamous cell. And the oxygen and carbon dioxide are exchanging across that very thin membranes there with just a little bit of basement membrane under each of those. Again, allows it for an almost instantaneous exchange of these gases. Again, they diffuse into that fluid at the alveoli. And this is part of the reason that we moisturize the air that's getting into our lungs so we don't dry out the alveoli. And again, the surfactant helping to keep that open. Sometimes when an infant is born way too early, one of the last things that develops in the lungs is this surfactant. Part of the reason that infants that are born really premature can have this respiratory distress issues because they're not making enough surfactant to inflate those alveoli in the lungs. So next time we'll look at some other stuff on this one, get a little bit more into the physiology.